Our next speaker is Dr. Anya Ermakova. She's a research scientist with Berkeley Psytech. She's a consulting in that role where she supports the clinical trial development and implementation. And she's currently working on a study in collaboration with King's College of London, looking into the safety and tolerability of 5-MeO-DMT. Dr. Ermakova holds a PhD in psychiatry from the University of Cambridge and a master's degree in conservation biology from Imperial College of London. Her areas of research interest include neuroscience and mental health, but also nature conservation and ethnobotany, all interweaved through the psychedelic science. On the conservation side, Anya is a member of the Council for Protection of Sacred Plants at the Chacruna Institute. Currently, she stays involved in both conservation and psychiatry research and hopes one day they can merge into one project. Anya previously worked at the forefront of psychedelic research as a science officer at the Berkeley Foundation and has provided psychedelic welfare and harm reduction services at PsyCare and Zendo since 2015. Please give a warm welcome to Dr. Ermakova.
Hello, good afternoon, everyone. It's such an honor and pleasure to present here for the first time in 2 years. Let me figure out how this works. Oh, not like that. Oh, yeah, today I'm going to zoom through a lot of topics about summarize the state of research about 5-MeO-DMT, talk a bit about history, natural occurrence, studies in animals, and research with humans. And if you want to know more about it, please check out the review that we wrote, just came out a couple of months ago. And I'm happy to talk about any of those topics in more detail in question and answer, Q&A part. Okay. Just a bit about my disclosures. I'm a consultant research scientist at Berkeley Psytech, and I work in collaboration with King's College London in 5-MeO-DMT trial. Oh, it goes backwards. Okay, now I figured it out. Sorry about that. The history of 5-MeO-DMT, it was first synthesized in 1938 and then isolated from plants in 1959 and subsequently been found in a variety of plants, although in very small quantities. Most notable of those plants are genera Adenanthera and Virola, known for their historical and ethnobotanical significance. And it's also been found in a couple of fungi, Amanita citrina and Amanita porphyria. But most well-known is occurrence in animals, and this handsome amphibian Sonoran desert toad, also known as Bufo alvarez. Colloquially, this is a very distinctive species. It's one of the largest toads it is the largest toad in the United States, and it's found in Sonoran Desert, as its name suggests. And from the glands, it's possible to express the bufotoxin, which toad uses for protection. It has very, very high percentage of 5-MeO-DMT, 15 to 30%, depending on the individual toad. It's also been found in mammals, in rodents, and in humans. The research in humans is inconsistent. It's found in some studies and not others, and a lot of those studies were done in the 60s and 70s, so we still don't know if there is endogenous 5-MeO-DMT and what role, if any, it has. In terms of legal status, 5-MeO-DMT is included in Schedule 1 Controlled Substance Act in the United States. It's also controlled in United Kingdom, Australia, New Zealand, and several other countries, but it's not listed by UN Convention on Psychotropic Substances, and various retreat centers operate using 5-MeO-DMT, either synthetic or from the toad. In terms of chemistry, I'm not going to pronounce its full name, but it's a tryptamine alkaloid similar to DMT and bufotenin. Pharmacokinetic research has been well characterized in rats and mice. It undergoes very fast metabolism, it's lipid soluble and easily crosses blood-brain barrier. It's barely detectable in blood 1 hour after administration. The main metabolic pathway is monoamine oxidase A, and there are other metabolic pathways that are involved to a smaller extent. In terms of receptor binding, it's non-selective agonist for serotonin and several other receptors. It has highest binding affinity to 1A and 1D receptors. But of course, we all know that receptor binding is just part of the story. We need to know a lot more about functional selectivity and secondary messaging cascades, and also, as Gul mentioned earlier today, to look at the epigenetic mechanism and what happens downstream after that. Behaviorally, in animals, it induces expected effects for other classic psychedelics, notably head twitch response, and also they discriminate 5-MeO from saline solution, but not from other psychedelics. There is also research on cardiovascular, thermoregulatory, nociceptive, and endocrine effects in animals. In terms of toxicity, sheep of all the species seem to be more susceptible to 5-MeO than and again, some animal studies indicate that drug interactions varying with lithium and monoamine oxidase inhibitors. In terms of human studies, there is for a long time there was mostly anecdotal evidence and reports of self-experimentation and some reports of the ceremonial use. There have been in the last 4 or 5 years epidemiological studies from people who use 5-MeO-DMT recreationally or ceremonially, as research by Alan Davis, Malin Zaug, and others. And ongoing now, there are listed 3 clinical trials, one at King's College London, the one I'm involved in, and 2 trials in Maastricht University. And one of them has just been published last week, and I'm going to talk about it in a second. In terms of trial at King's College London, it's a single ascending dose study, and it's also going to have a full pharmacokinetic profile and comprehensive safety research, and it's in healthy volunteers, so hopefully we could present results next time. And I'm doing also qualitative interviews with participants in this study in order to understand subjective effects better. People report that 5-MeO elicits more intense effects compared to other psychedelics. It's also very short, it has very fast onset and short duration of effects. It's also, people describe that it's very different from other psychedelics. There is often absence of visual effects, strong bodily sensations at first, but then people report not feeling their body, and people often report transcendent and mystical experiences involving non-dual awareness. And there is also increased range and intensity of emotions. Epidemiological research and various surveys indicate that there could be therapeutic benefits. There are some case studies and surveys from retreat centers where 5-MeO was administered alongside ibogaine, and there are also various prospective and retrospective surveys where there are noted improvements in mindfulness capacities, measures of depression and anxiety, usually self-reported increases in well-being. So from one of those surveys, most people report improvements in their symptoms associated with psychiatric conditions, and a very small minority report that it's worsened, and some people report no change. And this is the first interventional study, first clinical trial that's been published from Maastricht University. It was phase 1 dose-ranging study, and they had a single ascending dose and dose escalation arms. What they note is the very high inter-individual variability in response for 5-MeO, but generally they find that in those research settings, it's been well tolerated. There are not so many challenging experiences and effects, and adverse effects that were noted were very rare and resolved very quickly. So in conclusion, I want to say that 5-MeO is a short-lasting psychedelic with unique subjective effects profile. It has potentially high rates of ego dissolution and mystical experience, which, as we heard earlier today, correlate with therapeutic outcomes. Risk profile is broadly similar to other classic psychedelics, but we need to do more studies to understand it better. And we recommend same time of precautions to use with other classic psychedelics. And it's very promising for clinical exploration. And thank you very much, and I look forward to Q&A.